It's a pleasure to be able to introduce Professor Alan Cowman today. So many of you will know Alan is the head of the infection, and I'm going to get the name of the division wrong, sorry, seeing as I don't know any of these division names, the Infection and Immunity Division here at WEHI. He's also a visiting professor at Harvard. And so it might also interest you to know that he did his PhD here at WEHI before going to UC Berkeley and then returning here, and he's headed the division since 1999. So his work, as the title suggests, focuses on malaria, and in particular is looking at how the malaria pathogen invades the human red blood cells, how they remodel the red blood cells. But what's most relevant to today's talk is how the pathogen can invade the host immune system. And so by controlling the epigenetic, or by controlling the epigenetics, or by antigenic variation, these parasites can help to evade the host immune system. So clearly this is really important to understand, because if it's going to influence how vaccination strategies can be used. And all of his work has been extremely highly acclaimed, and he's received a very long string of awards throughout his career. He's a fellow of the Australian Academy of Science and a fellow of the Royal Society in the UK. So we very much look forward to your talk, Alan. Thank you.
Thank you very much. So I started getting this talk ready, and with two parasites in mind, Trypanosome brucei and also Plasmodium, which causes malaria. And realised there's actually quite a lot known about Plasmodium falciparum in particular, and perhaps even more than trypanosomes now, which is quite odd, since the work on trypanosomes really started a long time ago, and it's really only the last decade that understanding of antigenic variation, or the control of antigenic variation, has really come a fair way. So what I'm going to concentrate on is malaria, although I will mention trypanosomes just very briefly. So just first of all, an introduction into malaria for those who don't know anything about it. It's a major disease of humans, occurs mainly in tropical and subtropical regions of the world, and it's obviously relevant to Australia because a lot of countries just to the north of us have quite extensive levels of malaria and disease from malaria. So approximately 2 to 3 billion people are exposed each year, and 500 million people are infected. So at the moment, more than 700,000 die from malaria each year, and that's come down quite a lot. I used to have slides that used to say 3 million, so there's been a big effort to try and reduce that mortality, and that is working extremely well, although there's still a lot who die every year. There's currently no effective vaccine available, and drug resistance is a major problem. The parasite manages to develop resistance to to every drug that is thrown against it. This is one of Drew Berry's, in fact, this is the first animation that he made of malaria, and it's still my favourite. So this is part of the life cycle of Plasmodium falciparum. So people get it when Anopheles female mosquito bites, injects sporozoites, forms in, that then invades liver cells. So I've cut out the liver stage here, and this is the section of the blood cycle that I'm particularly interested in with respect to this talk. Merozoite forms invade the red blood cell, and they start this amazing remodeling process where they change this terminally differentiated cell into one in which the parasite can survive and hide from the host immune system. And part of this ability to hide from the immune system includes its ability to adhere to receptors on endothelial cells, plus also other cells, and this is critical to its survival during infection. Develops within this cell, and then bursts out, and new merozoites are able to invade red blood cells, and so that's the blood stage, or the asexual life cycle of the parasite. The ability to cytoadhere, as shown here in this, to the endothelial cells of this capillary, is mediated by a protein called Plasmodium falciparum erythrocyte membrane protein 1. And I should say that there are different species of Plasmodium that cause malaria, but I'll only be talking about Plasmodium falciparum, which is the one that causes virtually all of those deaths that I was talking about. And it's somewhat unique in that it, plus another Plasmodium, Plasmodium reichenowi, express this protein. The other Plasmodiums don't. So Plasmodium falciparum erythrocyte membrane protein 1 mediates this ability to cytoadhere to microvascular endothelial and also other cell types. And it does this by binding to particular receptors on these surfaces, depending on which particular PFMP1 type is being expressed at any one time. This is quite a large protein, can be over 300 kilodaltons long, so it's got to come all the way from the parasite through 3 membranes and then be deposited in this complex structure called knob structures. So the receptors that it can bind to include CD36, chondroitin sulfate A, and I'll talk about this one in particular, but also other receptors including ICAM1, VCAM, plus others. Now, PFMP1, as I said, protrudes from an infected red blood cell, and this is a scanning electron micrograph of an infected red blood cell. And you see these structures that are sticking out from the surface of the membrane called knobs. They're actually laid under the membrane, and they push the membrane up, and PFMP1 is deposited and concentrated within that knob structure. And that's absolutely critical to the site adherence function of the PFMP1 protein. And what we have here is just a mutant that we made that lacks these knobs. So this just shows the cytoadherence in action, where we have a slide here that is to which CD36 has been stuck. And so these particular Plasmodium falciparum infected red blood cells are able to adhere to CD36. And this is just comparing it to a mutant that lacks knobs, showing that the adherence is much less. So these parasites can adhere quite well under flow conditions that you would see in the microvasculature of the human host. And this flow, I think, is going in this direction. And you see the parasites in here. This flow is going in this direction. And you see the parasites within the red blood cell. There's 2 parasites within this one and 2 within this one. And you can see some of the contents of the parasite moving around under Brownian motion. Okay, so why does it express this protein called PFMP1 on the surface, and why is cytoadherence so important? And one of the reasons is firstly that it reduces splenic clearance. If it didn't cytoadhere and stick to endothelial cells, it would pass through the spleen and be fairly rapidly removed from the circulation and destroyed. That means that this protein, this very large protein, is exposed to the immune system. So this parasite, which is basically sitting inside a red blood cell, invisible, apparently invisible to the host immune system, has put this huge protein on the surface, essentially putting a red flag on it saying, here I am, come and get me. And so it has to have mechanisms to get around that, so it can evade the host immune system. And how does it do this? And the answer is antigenic variation. In other words, the parasite has different types of these PFMP1 molecules that are antigenically distinct and also have different cytoadherence properties, which I'll go into in a minute. So PFMP1 proteins are encoded by VAR genes, VAR for variable. Variable because they're hugely variable. The 60 are really quite distinct, but within the malaria population throughout the world, there's a huge amount of diversity in this particular gene family, and the parasite has mechanisms to generate this massive amount of diversity that it has. This protein family and the gene family that encodes this protein family are responsible, as I said, for clonal antigenic variation, for evading the host immune responses. There's approximately 60 genes per genome. So if every malaria parasite, Plasmodium falciparum parasite, has 60 VAR genes that encode 60 different PFMP1 types. As I said, a huge amount of diversity within a single genome, but also with much more within the population. And only one of these genes is expressed per parasite, a process called allelic exclusion, monoallelic expression, if you like. But basically, one is activated, the other 59 are silenced. And that switching rate that allows it to switch from one to one of the other 59 occurs at a rate of approximately 2%. So the switching is not being selected by the host immune system. It's an inbuilt mechanism in the parasite that allows it to switch at a very low rate. And that switching appears to be hardwired. In other words, it doesn't switch on in that 2% of the population all of the other 59 parasites. There appears to be a distinct order in terms of the expression. That makes a lot of sense. The parasite does not want to expose all of its PFMP1 molecules to the host immune system. It just wants to expose a very small number. So there'll be one that it expresses for the most part, and then there'll be a small subset of the population that is switched to another or another few PFMP1 types. And switching is controlled at the transcriptional level. There are no sequence changes, and this mechanism of antigenic variation is controlled epigenetically. Okay, so what advantages does it give the parasite beside splenic clearance? Firstly, this is just a good demonstration of antigenic variation, where you see switching from one population to another, or selection of one population to another. This is a cartoon of an individual with a malaria infection where they're originally infected, and this particular population expresses mainly one PFMP1 type. The immune system then basically mounts an immune response. The population is suppressed. There's a small number that express another type of PFMP1. That population grows. That's suppressed by the immune system as it develops an immune response to it. And again, you go through different waves of parasitemia, very typical Plasmodium falciparum infection. And you see the real thing here. And this is an individual looking at parasitemia, the number of parasites in the blood. And so you see these peaks and falls as the immune system gets on top of the PFMP1 type that is being expressed by these different populations. And as I said, PFMP1 is important for sequestration, which is absolutely critical for the ability of the parasite to prevent being passed through the spleen and being destroyed by that particular organ. But it has other functions as well. Firstly, the sequestration function and linkage to particular malaria disease phenotypes. Firstly, malaria in pregnant women is particularly linked to expression of a PFMP1 type that allows binding to chondroitin sulfate A, which is particularly rich in the placenta. So those parasites adhere in the placenta. That causes major issues with respect to the health of the fetus, but also the mother. And also cerebral malaria, particular PFMP1 types appear to be somewhat linked to this particular disease phenotype. For example, ICAM1 has been suggested to be important. So a PFMP1 type that binds to ICAM1 will tend to adhere more specifically within the brain. That can cause cerebral malaria, which causes coma and potentially death. But also there are other receptors, as I mentioned before, such as CD36, particularly associated with binding to endothelial cells by particular PFMP1 types. This particular protein allows what's called resetting, or one of the number of these PFMP1 types, resetting, which allows binding to red blood cells, probably is advantageous when the parasite releases its merozoites from this red cell, so it can invade these fairly rapidly before the immune system can get to them. But also, some can bind to particularly immune effector cells, such as dendritic cells, and these can affect the immune response. So it has a number of different functions which are absolutely critical to the survival of the malaria parasite. And so these different PFMP1 types are, as I said, expressed by different VAR genes. So what's happening is that in this population here, after the initial infection, you have mainly one PFMP1 type that's expressed, but within that population you have a number of other VAR genes that are expressing different PFMP1 types that are then selected by the immune response so that you get this different population. So you're selecting different populations that are expressing different VAR genes and different PFMP1 types. So VAR genes, as I said, there's approximately 60 per genome, and this is their location on the 14 chromosomes of Plasmodium falciparum, ranging from 800 kb to more than 3 megabases. So the genome's around about 23, 24 megabases long, divided into these 14 chromosomes. And the VAR genes are generally located at the ends of the chromosomes, as you can see here, these red arrows signifying location of VAR genes at the very end, at both ends. But there's some central clusters, particularly here on chromosome 4, for example, also chromosome 7 and chromosome 12. And so the parasite has basically developed a mechanism that allows it to shut down transcription of all of these VAR genes except one. Now, the different VAR genes have different promoter types, which is an interesting feature. And the promoter types that they have depends where they're localized in the particular genome. And when they're at the end of the chromosome, for example, facing in this direction from the telomere, they have what has been called an UPS-B type promoter. And if they're facing in this direction, they have an UPS-A, which is distinct from the UPS-B. When they're in the central clusters, they have what's called an UPS-C promoter. why? Not totally known. It's important for their regulation, and I'll mention that as I go through it. But some of these different promoter types have been linked to different disease phenotypes in malaria. And as I said, only one of these is transcribed per parasite. And this is a qT-PCR experiment. This is all 60 VAR genes for one particular parasite strain. And as you see, one is expressed and the others are fairly tightly regulated and silenced. So I'll talk about the mechanism that allows the parasite to silence these 59 and also to activate this one VAR gene and switch to another VAR gene type. Okay, so just with respect to VAR gene expression and the experimental tools that we have to really address this, this is looking at the transcription of a VAR gene within the blood cycle that I showed you at the beginning. As I said, these go from ring stages after the merozoite invades them. It's called the ring stage, trophozoite stage, schizont stage, and then new merozoites burst out and then reinvade new red blood cells. So they're just going through this cycle continuously. You know, they don't just express PFMP1 or the VAR gene and keep it on all the time and keep putting PFMP1 on the surface. They actually only express it for approximately the first 10 to 12 hours of this ring stage at the RNA level. Protein is expressed as well. It's exported to the red blood cell and actually stored, for the most part, within the red blood cell. It's only exposed on the surface of the red blood cell at about 16 to 18 hours post-invasion. So it's these stages that are able to cytoadhere, and these stages that can either interfere with the host immune responses or prevent passage through the spleen. So you have transcription at this point, and you'll have PFMP1 expression at this point, but at about 18 hours post-invasion, transcription is stopped, and this particular gene is silenced, but it's silenced in a particular way that's essentially called poise for transcription, because the parasite needs to be able to remember which VAR gene it had expressed in this generation, so that the next generation can express the same VAR gene. And as I said, it's important for the parasite not to show all 60 of its PFMP1 types to the immune system. It really needs to restrict it to a small number so that the host can't develop immunity to it fairly rapidly. So in terms of being able to analyze the mechanism of VAR gene regulation, we need a population that is expressing one single VAR gene type. And that's not easy when you have parasites growing in a culture dish. They're not under any selection. So it means that the VAR genes are being switched at 2% per generation. So if you start from a clone, you'll fairly rapidly get to a population that is expressing different VAR gene types. And this is an example here where a strain that has been grown in the laboratory, not selected at all, that is expressing different RNAs from different VAR genes within that population using a probe that's conserved between those different VAR genes. But if you select it, and the way we select it in this case is on a receptor, and we use in particular chondroitin sulfate A. So select for parasites that can bind to chondroitin sulfate A, that selects for expression of a PFMP1 type, that binds that receptor that is expressed by a single VAR gene. And you see what happens here, where you basically selected for expression of a single PFMP1 type and a single VAR gene. So what is the mechanism of allelic exclusion? How does the parasite manage to silence 59 of those genes and activate only one? And how does it move from activation to silencing, or silencing to activation? There are a number of different ways it can do this, and I said I'd briefly mention Trypanosoma brucei. And the mechanism for this one, although it does involve epigenetic control as well, is DNA rearrangements. That does not occur in Plasmodium falciparum. It does not involve DNA rearrangements at all. Although there's some similarities in Trypanosoma brucei, they have an expression, basically a position within the nucleus that allows a particular variant gene to be expressed, and that's similar, as I show you, with Plasmodium falciparum. Negative feedback, where the expressed protein can feed back to suppress the other genes, does not occur in Plasmodium falciparum. Non-coding RNAs do occur in Plasmodium and Plasmodium falciparum. Their role in control of epigenetic epigenetics and also control of VAR genes is not clear. There's some evidence that it is involved, but it's not really been analysed to any great extent. Nuclear compartmentalisation and gene repositioning, absolutely critical to silencing and switching, which I'll go into. And also chromatin structure, again, absolutely critical to the ability of the parasite to suppress and silence 59 of the VAR genes and activate 1. So the type of methodology we've used to look at the compartmentalisation of the nucleus and the position of an active VAR gene versus a silenced VAR gene, which is really what you need to understand with respect to how the parasite achieves allelic exclusion. Firstly, I think, which you all know, immunofluorescence assays, IFAs, using antibodies to particular nuclear compartments to work out where the compartments are with respect to others. But we've combined this with fluorescence in situ hybridization, FISH, basically labeling DNA or RNA with different fluorochromes so that we can work out the position of different genes within nucleus. In Plasmodium falciparum, the nucleus is actually very small. The parasite's very small. So this is a real challenge technically to be able to do compared to higher eukaryotes. So you can combine FISH with IFA, and I'll show you some figures that do this, where in particular IFA and DNA FISH, where we've combined detecting where a particular protein is and a position of a gene within the nucleus when it's either active or silenced. You can do that also with RNA FISH and IFA to determine when the position within the nucleus when it's transcribed, and also RNA DNA FISH. So this is a model of a general nucleus, and as I said, the nucleus of Plasmodium falciparum is much smaller, and the understanding of the compartments within Plasmodium falciparum of the nucleus is really not very well understood at this stage, but there are a number of things that are common. Firstly, positioning of heterochromatin around the nuclear periphery, which is present in higher eukaryotes, is also present in Plasmodium falciparum, and that's really important in terms of VAR gene regulation. And also, some of these other compartments shown here are really not been looked at all in Plasmodium falciparum. As I said, the nucleus is really small and it's really hard to do those sort of experiments. So the first thing that was found a number of years ago was that the end of the chromosomes of Plasmodium falciparum are actually at the nuclear periphery. And so this is a FISH fluorescence in situ hybridization with a probe called Rep20, which is within the telomeric ends right next to one of the VAR genes, and it's present on all of the chromosomes. So basically what you're looking at is all of the chromosomes, and first thing you don't see is 28 dots. And if you remember, I said there are 14 chromosomes. And what this says is that the 28 chromosome ends are actually clustered in the nuclear periphery in about 6 or 7 clusters. And the VAR genes are essentially the same, not surprisingly for the ones that are at the telomere ends, but also the central VAR genes are also clustered at the nuclear periphery, as shown by this FISH analysis here. So this is what the nucleus looks like with respect to the chromatin landscape, and not a lot different from higher eukaryotic nuclei, except it's somewhat smaller. And what you see is heterochromatin lining the nuclear periphery, but interestingly, you see basically a break, an area of what appears to be euchromatin, and it's euchromatin within the nucleus, but an area at the nuclear periphery that appears to be euchromatic as opposed to this more condensed heterochromatin around the nuclear periphery. And what I'll show you is that what we in other labs have shown is that the transcriptionally active VAR gene is actually positioned within this less condensed region, this eukaryotic region, whereas the clusters of chromosome ends and also the internal VAR clusters are actually embedded within this heterochromatic region in the nuclear periphery. But all VAR genes are at this nuclear periphery, whether they're in the end of a chromosome or in more central regions of a chromosome. And most, or all of them, except one, are basically packaged in heterochromatin. So the question we and others wanted to ask is, where is the active gene located within the nucleus, and does it change when you activate it from a silent state to an active state? And so for this, we use fluorescence in situ hybridization again, and the ability for us to select a VAR gene in an on state, and also select a VAR gene in an off state, the same VAR gene in an off state. So we could switch between on and off and ask, what position is that gene in the nucleus? And this is just one of the sorts of experiments that have been done to show that indeed VAR genes have to move from the heterochromatic nuclear periphery into that euchromatic region at the nuclear periphery. And essentially this is a VAR gene in a silent state, and we've marked it, I won't go into this in any detail, but we've marked it with a particular probe that we knew had to be in the active position, in the active site of the nuclear periphery. So when it's silent, they don't co-localize. When the VAR gene is active, in this case we use VAR2-CSA, which binds to chondroitin sulfate. So we could select on chondroitin sulfate A, then select on another particular receptor to switch it off. So we could go on and off as we liked. And so when it's active, it localizes with this marker for the expression site. And so this is basically a cartoon of what's happening here. We have the VAR2-CSA, which is expressing the PFMP1 that binds to chondroitin sulfate A. This is in the silent state, so we've selected it on another receptor. So it's basically embedded within the heterochromatin at the nuclear periphery. Another VAR gene has been activated. This is a particular marker that we use to tag the active site. When you switch it on, or select parasites that have it switched on using chondroitin sulfate A adherence, you can show that the active VAR gene co-localizes with this particular marker. So you need to move a VAR gene out of this heterochromatic region into this more transcriptionally permissive region for it to be activated. And we know that that requires actin polymerization, so actin polymerization is involved in this relocation from a repressive to an active site at the nuclear periphery. But it's not as simple as that, because if you remember the VAR genes that I showed you, there's more than one VAR gene at these ends in many cases. And so if you're moving the whole end of a chromosome into this more permissive transcriptional area, you may well be moving a number of VAR genes in. So we don't know what's happening there. It may be that one VAR gene loops into this region and the others are kept in this more heterochromatic region in a silent state, or they're put into this region but somehow altered so that they're not transcriptionally active. We do know that P. falciparum undergoes closed mitosis, and so the chromosomes do not condense during nuclear division. And this means that the localization of VAR genes in the nucleus can be maintained and can be transmitted to the next generation. So what this means is that the epigenetic inheritance of the particular VAR gene that's being expressed basically can be passed on. So this is important. I'll come back to that later on as well. Okay, so what about chromatin modifying proteins? We know that an active VAR gene is an open chromatin structure, and that's been shown. We also know that silenced VAR genes are in a closed chromatin structure. In other words, they're in heterochromatin, whereas an active one is euchromatin form. With respect to the regulation of movement from euchromatin to heterochromatic forms, we know that histone code is absolutely critical. And you may have gone through this before, but I'll just briefly mention that the basic unit of chromatin is the nucleosome, which is basically 8 histone molecules that package the DNA into a unit that can be packaged into a higher order chromatin. Within this these histone molecules, the N-terminal so-called tail is protruding from the nucleosome, so that these can be modified by chromatin modifying proteins. And this is absolutely critical to regulation of chromatin structure and also the regulation of transcription. So chromatin can move from transcriptionally competent euchromatic state to a condensed chromatin, heterochromatin form. And these marks on histones appear to be important in this. And although at the moment, at least for us, they essentially mark the more open state versus the more closed state. And these different modifications can involve a number of different groups, including acetylation and methylation, ubiquination, simulation, and phosphorylation. I'll only talk about these two, acetylation and methylation, because they're the main ones that are being concentrated on in Plasmodium falciparum. And I'll particularly talk about histone 3 with respect to the different proteins that modify it, that affect VAR gene expression in Plasmodium falciparum. And the function of these histone modifications are to recruit non-histoproteins to regulate the state of this chromatin and also transcriptional state of close by genes. And these bind particular modifications on the N-terminal tails of histones, such as chromodomain here, which will bind methylated lysine on histone. I'll talk about a chromodomain protein, as well as a PhD protein, containing protein. And the methods that I'll be talking about, again, you probably know this and have had this described previously, is essentially chromatin immunoprecipitation, or CHIP. In this, we're able to purify nuclei, then sonicate the chromatin to small pieces. We can then immunoprecipitate particular chromatin subpopulations using antibodies to either proteins that are bound to the chromatin to look at the landscape of binding across the whole chromosome, or to different modifications on the different histones, to purify DNA, and then to analyze them using different methods. And I'll talk particularly about ChIP-ChIP, which is using microarrays to detect which DNA molecules are particularly coming down with these immunoprecipitations, but also ChIP-seq. Firstly, histone silent information regulator 2, which was discovered back in yeast quite a long time ago, and was important for regulation of yeast mating type locus. And silencing information regulators 2 are histone deacetylases, and they act in concert with histone acetylases to balance the level of acetylation of the chromatin. So a more acetylated form is basically linked to euchromatic and open form, in other words, a transcriptionally active form, and a less acetylated form is linked to heterochromatin and more silenced forms. So one of the first questions when we started looking at regulation, epigenetic regulation of Plasmodium falciparum VAR genes was, are there any SIR proteins, silencing information regulator proteins, in Plasmodium falciparum? And the answer is there are indeed, and there are 2. We call them PSSIR2A and PSSIR2B. Both genes have been disrupted. It's not lethal, so the parasite can survive without both of these. But the interesting thing about this, when you disrupt one or the other, is that it activates many VAR genes. In other words, you release the silencing of a lot of the VAR genes, and you end up with multiple VAR genes expressed and multiple PFMP1 proteins on the surface of the red blood cell. And you can show this quite clearly here in this experiment, where we've selected for VAR2CSA on and then knocked out the SIR2A gene. And this is the parental line expressing one single VAR gene, in fact the VAR2CSA gene. And when you knock out SIR2A, you see a large number of VAR transcripts. So the removal of SIR2A alleviates VAR gene allelic exclusion. So the SIR2A protein and also the SIR2B protein, I won't show you the data, but SIR2B protein also does a similar thing but to a different subset of VAR genes. And this is just a microarray to show that where, so all of the genes that aren't altered in this SIR2 knockout in this example are essentially on this here, on this line, whereas any that are upregulated are shown in either green or red, and these are mostly VAR genes. There's some other genes that are actually closely linked to VAR genes that are also epigenetically controlled, and some of these are released from their silencing mode. Not all in this SIR2A knockout, but if you do the SIR2B knockout, you see all these that are particularly silenced in this case are activated, and all these that are activated in the SIR2A knockout remain silenced. So between the two of them, they essentially are required for silencing of all VAR genes. SIR2A has pleiotropic effects, and you can see that here, and this is just looking at the length of chromosome ends of different parasite lines, the wild-type parental line. So SIR2A knockout, and you see much longer chromosome ends, telomere ends, compared to SIR2B, which isn't altered from wild-type. SIR2A also associates with the nucleolus and regulates the level of RNA transcription. So it clearly has other effects other than the regulation of VAR genes. Okay, so the second protein I'll talk about is heterochromatin protein 1. It's a highly conserved adapter protein, important in gene repression. It has a chromodomain, and as I said, that binds to a methylated lysine. It is involved in heterochromatin formation and stability, and in sequestration of genes to the nuclear periphery. And there is one heterochromatin protein 1 in P. falciparum. So at the moment, you can't knock that gene out. It appears to be lethal. Plasmodium falciparum is a haploid. Basically, if the gene's essential, you essentially can't knock it out. And people are trying to knock it down to look at its particular function. But what I have here is basically the localization of heterochromatin protein 1 on the chromosome, the whole genome of Plasmodium falciparum, as analyzed by chip-on-chip. This is done in Till Voss's lab. And starting from chromosome 1 to chromosome 14, you see, first of all, that the position of heterochromatin protein 1 is in the telomeric regions and subtelomeric regions. And if you remember where VAR genes are, they're basically localized, particularly at the ends of each chromosome, but they're also in the middle. And if you look at the central regions of the positioning of heterochromatin 1, you see that they're exactly overlaying VAR genes as well on chromosome 4, for example, chromosome 7 here, and chromosome 12 here, again, the two clusters of VAR genes. So PFHP1 demarcates the subtelomeric region and also the position of VAR genes throughout the genome, and has to be important in the repression of VAR gene transcription. Just to show you that the marks, so as I said before, these marks on different histones, such as histone 3, lysine 9 methylation, appear to be, or at least mark different transcriptional states of genes. Till Voss's group also mapped the position of this particular mark with respect to PFHP1. And this is just looking at chromosome 12 and the central clusters versus chromosome 10, positioning of HP1, and the VAR genes are in green, and you see it exactly overlays the VAR gene in this case, as does this particular mark on histone 3, histone, the K9 methyl 3 mark. So it suggests that HP1 probably binds to this particular mark, although that has not been shown directly. HP1 positioning here, this is a subtelomeric or chromosome end, one VAR gene in this case, and you see that the positioning of HP1 and also this particular mark is a bit broader and covers not only VAR genes but some of these other virulence genes that are also epigenetically regulated. So we know that these particular chromosome marks are important, or at least allow us to map the state of VAR genes. For a sans VAR gene, we know that for histone 3, lysine 9 methylation is basically linked to establishment and maintenance of silencing. Absolutely. You can see that every single VAR gene is covered with chromatin that is marked with this particular methylation. For the active VAR gene, we know that not only does it have to be at the nuclear periphery in that euchromatic area, but it has to be marked with these different marks on particularly histone 3. Histone 3 lysine 9 acetylation is linked with activation of the VAR gene, and histone 3 lysine 4 methylation is also, particularly in the promoter region, is a mark that is important for active VAR gene transcription. In the poised state, which, as I said, is basically the state between when it's expressed from one generation to the next generation, you find that, in particular, histone 3 K4 methyl 3 is important, and it's also important for activation of gene expression. So we knew that there was an expression site in Plasmodium falciparum, and to really get into that and start to understand what proteins were important for that, we needed to start to identify proteins within that spot. And we basically had started a program, Jennifer Volz in the lab, to identify candidate proteins involved in epigenetic regulation within this expression area of the active VAR gene. And we did this by basically looking at all of the proteins and genes that we thought would be involved in epigenetic regulation, tagging them with GFP or HA, and to identify any that were expressed and localized within that single spot. And out of the 30 that were looked at, one was found, and that happens to be a P. falciparum histone lysine methyltransferase, that we call PFSET10. It resides in the nuclear periphery, and you can see here this basically is an IFA, antibodies to this particular protein, PFSET1, and you see this single spot in the periphery of these two nuclei. So this is two parasites within the one red blood cell. So one protein so far that clearly appears to be expressed within this area of the nuclear periphery. And you can compare that with heterochromatin protein 1. As I said, it is basically linked with all of the silenced VAR genes, and so you see the other clusters, but it does not co-localize with PFSET10 antibodies. So this appears to mark the transcriptionally active VAR gene, whereas PF heterochromatin protein 1 marks the other clusters that are silenced. So the question was, does PFSET10 localize to the active VAR gene? And I'm not going to go into this in detail, but basically what it is IFA fish. So basically we're localizing the position of SET10 with the position of a VAR gene, in this case VAR2CSA, which expresses the PFMP1 that adheres to chondroitin sulfate A. So we can have this particular gene in an off state and an on state, and we can localize the position of PFSET10 spot with respect to that. When VAR2CSA is off, you see no co-localization of VAR2CSA with the silenced VAR2CSA with PFSET10 protein. However, when it's on, you see co-localization or localization in close proximity to PFSET10 and the active transcribed VAR2CSA. And that's in stark contrast to heterochromatin protein 1, where you have here VAR2CSA in the off state, so it should be in the heterochromatin. Heterochromatin protein 1 marks the heterochromatin and the silenced VAR genes, as I've shown you, and you see that basically it is in close proximity to PFHP1. This is VAR2CSA in the on state, and so you've marked all of the other chromosome clusters or VAR gene clusters, and you see no co-localization. So PFSET10 does localize to the active VAR gene. What is PFSET10? And I've already told you, it's a histone 3 lysine 4 methyltransferase. In particular, the SET domain, we've shown that it is active, and it's particularly methylates histone 3 on the lysine position. It has a PhD finger as well, and PhD fingers can bind to histone 3 trimethylated lysine 4 marks. So this particular protein is a histone 3 lysine 4 methyltransferase. What is it actually doing? And I won't go into the data that suggests what it's doing. What I'll do is just describe what we believe it's doing. Firstly, it's expressed during a poised state of the active VAR gene. So it's not expressed during the transcription of the active VAR gene, but it comes on as that transcription stops. And then it remains on whilst that it's in its poised state. It's associated with the expression site of an active VAR gene, so it's localized as a single dot at the nuclear periphery of a nucleus. And it maintains the active VAR gene in a poised state. And it's important for memory, for heritable transmission of essentially that VAR gene activation to the daughter cells, so that they can remember that this VAR gene is the one that was expressed in the next generation. So this just is a bit of a summary, mainly for the regulation of PFZ10, but it does have other aspects on it which are important. So the first layer of regulation of VAR gene silencing and activation is positioning within the nucleus. We know that all VAR genes are positioned at the nuclear periphery, that all but one are positioned within the heterochromatic portion of that nuclear periphery and silenced. One is within the eukaryotic region and active, and that in order for it to be silenced, it has to move into this region here. In order for one to be activated, it has to move into this region here, and that involves actin polymerization. Activation, we know that the methyl mark on lysine 4 of histone 3 is particularly important. Specialized histones such as 2HAZ are also involved. I haven't gone into that, but work from Mike Duffy. Duffy's laboratory has shown that they are involved in this activation process. Also, lysine 9 of histone 3 acetylation is also important with respect to activation. Silencing, both SIR2A and SIR2B are important for deacetylation so that it can be packaged into a heterochromatin and silenced. In terms of the poised state, we know that PFZ10 is important. It is a histone 3 lysine 4 methylase, methyltransferase, and can basically transfer methyl groups to this N-terminal tail of histone 3. We also know that the acetyl mark on K9 is important with respect to this. In terms of silencing, we know that deacetylation is important via SIR2A and B, that heterochromatin protein 1 binding probably to K9 lysine 4 methyl 3 is important for maintenance of this silent state, that histone lysine methyltransferases are important for methylation of this to keep these in the silent state. And I haven't talked about this factor here, which is also important in terms of silencing and activation. So in summary, PFMP1 is the major virulence protein in Plasmodium falciparum. It's responsible for cytoadherence to host cells, and that exposes it to the host immune system, so the parasite has a mechanism of antigenic variation to basically vary the expression and also the different antigenic type within a single infection and also within the larger population. It's epigenetically regulated. We know that VAR gene positioning to a heterochromatin-free domain at the nuclear periphery is required for activation, and that PFSIR2A and B, which are histone deacetylases, are important in silencing of VAR genes, and also that heterochromatin protein 1 is also important for maintenance of VAR gene silencing. And also that PFZ10, which is a histone 3K4 methyltransferase, is important for activation and heritability of an active VAR gene in the poised state. And we believe it maintains a poised VAR promoter state so that it can be remembered in the next generation. And that's absolutely critical for the ability of the parasite to remember. The last thing it wants is to express all of the VAR genes or even a subset of the VAR genes at any one time. Okay. And I've mentioned quite a lot of work from a number of different laboratories, but the people at WEHI who have been involved in this work, particularly Jennifer Volz for the SET10 work, but also, sorry about this, but also Till Voss, Manos Jurasingh, Till Voss and Chris Tonkin with respect to particularly SIR2B for Chris. Also a lot of work from Henk Stunnenberg's lab in terms of the chip. I briefly mentioned some work from Mike Duffy. So thank you very much.
For a great talk. So we'll start off with student questions.
From in the back.
Is there anything known about how the next VAR gene that will be activated is chosen?
Short answer is no. It's been looked at as to, with respect to one line in particular, which VAR gene comes on and what's the order of VAR genes that come on. And the answer is in that case that it tends to be the same every time. So if you clone it out and then basically allow it to switch to other VAR genes with no selection, it'll switch to the same one and then the same one after that. So the order appears to be hardwired. Exactly what that mechanism is, we don't know at this stage.
So I might have misunderstood this, but in the chart where you were showing the areas of chromosomes which HP1 associates with, and maintaining heterochromatin at stage. So I would have expected the asexual stage would be suppressing a whole lot of genes in heterochromatin, like anything into homosexual stages, for example. So are they not maintained in heterochromatin, or are they not maintained by HP1, or am I misinterpreting that graph?
They don't appear to be maintained in heterochromatin, and they appear to be under basically transcriptional control. There are transcription factors that appear to be important in activating those. Even within the asexual stage, there's a lot of genes that are basically silent up until a point within that cycle. They don't appear to be within the heterochromatin. There are some, as riffin genes, stevedours, appear to be in heterochromatin and epigenetically silenced. There are others, such as invasion genes, EBA proteins, perhaps even the RH proteins, appear to be within the heterochromatin and may well be epigenetically regulated as well. And there is some data for EBA140, for example, for that. For the most part, they're in euchromatin, and even when they're not transcribed, they're in euchromatin. They appear to be under transcriptional factor control.
Is that considered unusual? I mean, in terms of the genes are actually being used during that life cycle, they're the ones that are being stored in what you would think is being more kind of long-term storage format, whereas the ones that aren't being required at all for a long time, that are required much further down the life cycle, they're maintained by transcriptional regulation.
Is it unusual? I thought it was unusual for the invasion genes, not all the main genes, the one EBAs and RNAs in particular, to be in heterochromatin and to be epigenetically regulated rather than what I thought would be the way they'd be regulated, and that is essentially be under transcriptional control of expression of a transcription factor. So I guess I have the opposite view. No, I don't think it's unusual.
So would destabilising the epigenetic regulators be a possible therapeutic target now? Is that why you're going along these lines?
Yeah, there are some people who are developing compound, or at least testing compounds that have been developed, particularly anti-cancer compounds. For example, that hit acetylation targets of proteins, and they work quite well against Plasmodium falciparum. So there are people who are trying to develop those sorts of compounds as anti-malarials.
Any other questions?
Just a quick one, just more of a background one. You showed that the infected red blood cells, when they're expressing the PFABs, can bind to...
When they're expressing PFAB1.
So when the PFE-MP1 is on the surface, they can then bind to, was it CD36 on dendritic cells? What's the reason for that? Talking about parasite. So
there's some evidence that suggests that binding of PFE-MP1 to CD36 on dendritic cells alters their function. There's a number of papers that basically describe that from Peter Urban's lab. And so there's some data suggesting that PFE-MP1s are important for regulating immune effector cells and altering the immune response. So there's some data from here with respect to PFE-MP1 regulating function of NK cells from Louis Schofield's laboratory. So there's reasonably good evidence that it is doing something with respect to altering host responses.
Let's just say, because I think we're out of time now.